inviting me, even though I'm from Kansas State, you know, it's a little bit north. Anyway, you beat us in football games, so we're all good. Today I'll be talking about, again, my animal model I use to look at the effects of early environmental enrichment on drug-taking behavior in adulthood. And before I get started, I need to acknowledge the people that make this work possible. At Kansas State, I collaborate with two faculty members, Dr. Stephen Kiefer in the Department of Psychological Sciences, Dr. Stefan Bosman in the Department of Chemistry, and then a host of former graduate students, current graduate students, and wonderful undergraduates. My former graduate student, Eric Garcia, is now a postdoc at UT Galveston, and then I have a great team of undergraduates and graduate students in my laboratory. And my research is supported by the National Institute of Drug Abuse, and then a Cobrae Center at Kansas State University, the CNAP Center, with a focus on cognitive neurobiological approaches to plasticity. So thanks to those sources for my funding. So to look at the effects of the early environment on drug-taking behavior in adulthood, I use a very well-established model of altering the early environment. And that's a model of differential rearing, and this model was actually established back in the 1960s. And this is a picture from the New York Times in 2017, but it's from 1964. And you can see, I don't want to get you with the laser, but you can see, sorry, I'll step back here. Dr. Rosenwig, Dr. Diamond, and Dr. Bennett looking in at their enriched environment. And they're one of the first teams to demonstrate that changing the early environment for rodents changed, altered learning and memory, and altered brain function in adulthood. And their early model was picked up by a lot of other researchers. Those of you familiar with this work may have heard of another researcher, Greenow, did a lot of work in this area. And enriching that early environment significantly improves learning and memory and improves synaptic plasticity. And this work has also been extended beyond learning and memory into recovery of function. So providing environmental enrichment helps with recovery of function from stroke, recovery of function from traumatic brain injury, and it's even been used, I focus on the early environment, as did they, early life, but it also helps with older adults, environmental enrichment. It's exploded, this area of research, from the early 60s. And my focus in using this model is looking at how does this early environment alter drug-taking behavior. That's what my group and several other groups are interested in. So this is 1964. A good deal has changed in terms of regulations for animal research since 1964. So this is our enriched environment in 2019. But we follow the same core tenets, just a lot has changed. So we use a variety of novel objects that are changed daily in the cage. The animals are housed in a large group. They have to have a cohort of anywhere from 8 to 14 animals, so they need that social contact with other animals. And then they're handled daily, every day, by us, the experimenters. So novel objects, daily handling, and contact with their cohort. They're placed in these environments at 21 days of age, which for a rat is right after they wean, and they remain in the environments until late adolescence, early adulthood. And one advantage of an animal model, that's only 30 days. So we place them in the environments, boom, 30 days later we're in late adolescence, early adulthood, and then we can start training and testing them. So we have a rapid developmental period. This environment is in stark contrast to the isolated environment. This, if any of you again are familiar with these models, these used to be the standard of care, old hanging wire metal cages. They have no contact with us for 30 days. We place them in, and if we see no adverse health outcomes, we monitor their food and water intake every day. If we see no adverse health outcomes, we don't touch them for 30 days. They have no novel objects and no contact with a cohort, no contact with other animals. So it's a stark contrast from the enriched environment. And those of you, again, familiar with this literature, this is also sometimes called an impoverished environment, and I'm happy to talk about why we call it isolated and not impoverished. Anyway, so we use this enriched, isolated environment, and then one of the challenges in this field is what is your control group? These two groups differ in terms of cage size, bedding, novelty, cohort. So what we do and several other groups do is we just use a standard put forth by the National Institute of Health, and we have 2 rats per cage that are handled once a week. It's sort of, what does the average Joe rat do? Okay, what's the average rat doing in the lab? This is a standard of care across the country. We do not control for all the differences between the 2 environments. We compare our 2 environments to the average laboratory environment. And then what we do is we ask the question, how do these environments during early childhood alter drug-taking behavior in late adolescence, early adulthood, later adulthood? We can ask throughout the lifespan after that. The advantage of using, there are a lot of advantages to using animals for drug use research. One of the great things we can do is we can look at the onset of drug use. We cannot look at onset of drug use in humans. We cannot give adolescents drugs for the first time for obvious ethical issues. And then working with human drug users, many of them engage in polydrug abuse. Many of them are not maintaining their health and nutritional aspects. So with animals, we can tightly control those health and nutritional aspects and just isolate the drug effects. So animal research and drug abuse offers a lot of advantages as a model system for us. So the way we ask animals about drugs, there are a lot of ways we can actually do this. I'll take you back to your psychology classes in college here. We use an operant chamber or a Skinner box. Our rats have levers they can press. And then we use some type of cue light or tones for Pavlovian conditioning. And then they can earn a food reward, sucrose pellets, sugar, something sweet to drink, alcohol to drink. We can give them a lot of different things to earn in the operant box. And what we're usually giving them in the operant box in my lab is they actually get an intravenous infusion of a drug that we're studying. We implant an indwelling jugular catheter using surgery. And once they recover from surgery, about a week later, we place them in the chambers and they learn to lever press for an infusion of the drug. So this tubing connects to their catheter. And then in the pump, we have the drug that we're studying. They press the levers in the schedule we set. And then we often have condition cues that come on with the drug infusion. So we can look at Pavlovian condition factors also. And most of the time what we're doing is we're manipulating some environmental or pharmacological factor. We're seeing how it alters drug taking, and we measure that drug taking through the lever press behavior of the rat. This is a good model of drug taking in terms of the animal must choose to lever press. Much like a human has to choose to take the drug, they have to choose to lever press. And I'll give a little bit of a spoiler. Our enrichment is protective against drug taking. It's so protective, our enriched rats will often go to sleep. So they definitely have choice in this matter, much like humans have a choice. So when compared to isolated condition rats, enriched condition rats self-administer less psychostimulants. Psychostimulants include drugs like cocaine, methamphetamine, and amphetamine. They also self-administer less of alcohol, nicotine, opiates. I'm just going to focus on psychostimulants for the sake of brevity today, but across all drug classes, it's reducing drug taking. And self-administer means they're lever pressing to get less of those IV infusions. And we see the most robust effect with low unit doses of psychostimulants. As we increase that dose, the enrichment is less protective. So to show you some of what does that data look like, this is increasing doses of amphetamine here. This is the number of infusions the animals earn during a 1-hour session. These are our isolated rats in blue, the enriched rats in red. And at these lower doses, the enriched rats are self-administering less of the drug. And then as we increase the dose of amphetamine, that enrichment effect, it's just no longer protective. And if you're interested, the S here on the graph is saline. We actually had some days where we just substituted in saline. They were lever pressing for no drug, and the isolated rats were still lever pressing just for a saline substitution, not even for a drug. To show you more at that higher dose where the enrichment is not protective, these are repeated daily 1-hour sessions where they were given a high dose of amphetamine, 0.1 milligram per kilogram. That's a very high dose of amphetamine. And you'll see there are no group differences. So enrichment no longer protects with those higher doses. So one of our questions was perhaps, we don't see increased vulnerability in the isolated rats here either. And one of our questions was, if we extend the session length out, would enrichment be protective? Would isolated rats be more vulnerable? And researchers in the field are moving to longer sessions to better model human drug-taking behavior. Humans taking psychostimulants often binge for a period of 3 to 5 days, take a break, binge 3 to 5 days, take a break. So they're moving to longer sessions of binge model. And what you'll see is that the animals have an escalating increase in drug-taking behavior. You'll see it's called compulsive drug-taking. So we tried 6-hour sessions. They had 3-4 days on, take a break, 3-4 days on, take a break. And we did that for 12 sessions. And we wanted to know, would enrichment be protective? These studies are ongoing, but we have 12 sessions at 6 hours each. and infusions earned. First of all, just notice how much more amphetamine they're earning across a 6-hour session. And we are seeing that enrichment is protecting against the escalation of drug taking across the binge pattern across the 6 hours, which is exciting. We are not seeing increased vulnerability in the IC rats, so it looks like just enrichment is protective. We're not seeing increased vulnerability. So enrichment does protect against the intake of psychostimulants. And I think as many people in this room know, another really important phase of drug use that we need to examine and explore and look for protective factors is relapse. Unfortunately, many individuals go to treatment facilities and get help for their drug taking, and when they return home, they relapse. And some estimates are upwards of 60 to 80% of people will relapse. And one of the causes of that relapse is believed to be Pavlovian conditioned cues in the home environment that trigger the relapse. So in an animal, again, this is relatively easy to study. We give them extended access to the drug, and then we expose them to the Pavlovian conditioned cues after some break period. So in a relapse test in an animal, we put them back in the chamber where they previously got the drug, and they're exposed to the conditioned cues previously associated with the drug in the conditioned chamber, but they don't get any drug. We put saline in the pump, and they're lever pressing for nothing. Again, advantage of an animal model. So at the end of training, just to show you, this was with 1-hour sessions. There were no differences between our animals and their lever press behavior. They had a 10-day break. And then we looked to see just how much would they lever press for those cues previously associated with the drug. How much would they relapse in the presence of the cues? And we saw that enrichment, in red, protected against a relapse. The isolated rats had the most lever pressing, and then the average rats also had an increase in lever pressing, but the isolated were the most vulnerable to relapse. So our hypothesis of all of this is, I do a good deal of behavioral work. I'm also a neuroscientist. Our hypothesis is that this differential rearing is altering the functioning of a neurotransmitter called glutamate. It's important for learning and memory, all that work in the 60s. It's also important for drug use. And we hypothesize that isolated rats have a dysfunction in their glutamate, and enriched rats have enhancements in glutamate function. So what we're working on now is we are doing these long access sessions. We're giving them extended access to amphetamine. We're then giving them a break. But during that break, we're giving them a drug called N-acetylcysteine. And N-acetylcysteine you can buy at your local health food store. It's available over the counter at your local GNC. And it's a cysteine prodrug. It apparently, if you have friends who are like into bodybuilding, it helps promote muscle growth. It helps with free radicals, but it also, for us, importantly, reduces drug craving. And there's been some recent studies with humans that it reduces craving for cocaine, and one recent study where it reduces craving for methamphetamine. It also alters glutamate function. So during the break period, during the abstinence period, we gave the animals N-acetylcysteine with the hope it would restore glutamate function in our isolated rats and reduce that vulnerability to relapse. And then we gave them the relapse test, and then I won't show this data, but we look at a bunch of proteins of interest in different brain regions important for controlling the response to glutamate. And what we observed, this is in the group not treated with the N-acetylcysteine. The enrichment with this is significantly protected against the relapse compared to the isolated and standard housed rats. And the N-acetylcysteine decreased the relapse specifically in the vulnerable group, in those isolated rats. So this is really encouraging data, but this study is still ongoing. We're still exploring those proteins to ensure these effects are due to the neurotransmitter glutamate. So to summarize, differential rearing results in changes in drug taking and relapse. It's protective for enriched rats. It enhances vulnerability in some measures, not all measures, for our isolated rats. And we're hypothesizing that these differences in vulnerability are due to changes in functioning of the neurotransmitter glutamate, or what we call glutamate homeostasis. So thank you for listening to a talk about rats and preclinical work with drug abuse. I appreciate your attention.